Hello and welcome dear viewers to the another topic on dissolution, and in this video we are going to uh, see what is dissolution similarity and the dissolution similarity requirements for generic product development. So as we know that dissolution is a performance test for the formulation, and dissolution has a great impact onto the generic product approval and the bioequivalence requirements. So see, what is generic product and what is reference product. In basic we will see the reference product and generic product. So reference product or innovator product, it is also known as RLD or comparator product. It is the product which is approved for the first time by the regulatory authorities, and generally these type of products are novel. They are protected by the patents, and these products may be made by using the new chemical entities or the new formulation designs. Then generic product is the product which is as similar to the innovator product in terms of the bioequivalence and the stability requirements. Then the safety and efficacy of the generic product should be in line to the reference product. Before going to the bioequivalence study for generic product, the generic applicant performs the dissolution studies including the multimedia dissolution and bio-relevant dissolution testing as per the product requirement, and these testings are done to know the behavior of the reference and the test product in vitro. So dissolution test is the in vitro test, and this test can guide the generic applicant for selection of the batches for pivotal or pilot bioequivalence study, and to understand the potential differences between the two formulations, so that the dissolution test becomes very crucial. Then dissolution testing is a product performance test, and dissolution can influence the drug's bioavailability and the therapeutic efficacy. Dissolution testing and profile comparison is a key requirement for the product approval, and also it can reduce the cost of bioequivalence study to prove the safety and efficacy. So you might be knowing about the BCS based bioavailability and the bioavailability for the lower strengths. If you are going to perform the bioequivalence study with reference product on higher strength or the strength which is critical for bio, and then the other strength will get the biowaiver, so that biowaiver is based on the dissolution similarity. For getting the biowaiver, for getting the product approved from the regulatory bodies, the dissolution similarity is required to be established. Dissolution similarity applications. If you show the dissolution similarity, then what are the applications for that dissolution similarity? First is BCS based biowaivers are based on the dissolution similarity. Then biowaiver for additional strength are also granted by the regulatory authorities based on the dissolution similarity. If the applicant of the generic formulation fails to prove the dissolution similarity, then the regulatory body will not give the approval. Dissolution similarity provides the information for selection of the formulation strategy to go ahead for the bioequivalence study. See, the dissolution testing and dissolution similarity starts from the R&D, scale up, technology transfer, commercial production till the life cycle. So if the product is getting manufactured at some site and it is required to be transferred to other site, that time also dissolution similarity is required to be established. So dissolution is the key performance test. Then SUPAC changes and product variations, whether you are going to do the variation in the composition, manufacturing process or site, then these variations are approved on the basis of the dissolution similarity. The formulations with dissolution similarity are considered as bioequivalent. Then if you are using the score tablets or tablets with break line, that time the score tablet dissolution similarity is required of the whole tablet with the half portion of the tablet. So these are the some dissolution similarity applications. Now we are going to the approaches to prove the dissolution similarity. See, you and me cannot say that the dissolution is similar or it is not similar. There are certain requirements, there are certain calculations, and there are certain scientific approaches for establishing or for showing the dissolution similarity. So model independent approach and model dependent approaches are there. We will not go in detail for these approaches. We will see the dissolution similarity on model independent approach only, and in that we will see the F2. So model independent approach involves the similarity factor, that is F2. Model independent multivariate confidence region procedure, this is another technique or approach, and multivariate statistical distance. So this is another method for providing the rationale or providing the justification for dissolution similarity. Then model dependent approaches are linear, quadratic, logistic, probit and Weibull models. So these are the different models which can be used to show the dissolution similarity. Now we will see one by one. So dissolution similarity when it is not required means calculation or the statistical calculation requirement is not there. When the test formulation and the reference formulation, both the product shows the percentage of drug release or drug dissolved is more than 85% within the 15 minutes. If you are working on a highly soluble molecule or you are working on the low soluble molecule, but if its dose is less and you get dissolution of above more or more than 85% within 15 minutes for test and reference product, that time you are not required to prove the dissolution similarity. No mathematical calculation is required and dissolution is considered as the same or similar. And if you consider the US and Europe requirements, then this is similar for US and the Europe as per the guidelines and as per the requirements. So example I have given here, you can see this example. In 15 minutes, reference product is showing 88%, test 1 formulation is showing 90%, test 2 is showing 87%, test 3 is showing 87%. So these three formulations or three approaches you can consider, or the three dissolution profiles you can consider, and all these are giving the dissolution more than 85% within 15 minutes. That's why these can be considered as similar. And the reason behind these 15 minutes might be that the formulation shows dissolution or complete release more than 85% of the release within 15 minutes, and 15 to 20 minutes is considered as a gastric emptying time. So in this case it can be considered or understood that if the formulation shows very rapid release, then the formulation behaves like a solution and dissolution will not be a rate limiting step. That's why the dissolution similarity calculation is not required and the profiles are considered as same. This type of dissolution similarity is called as point comparison. So point comparison means you are considering only one point of 15 minutes and you are showing the similarity in the dissolution. So always remember, if you have a formulation which shows above 85% drug release within 15 minutes, then there is no requirement of F2 calculation. Also remember that these rapid release of more than 85% in 15 minutes should be there in all the medias. Then the second will be when the dissolution profile is considered as similar, it is second. If F2 value between 50 and 100 suggests that the two dissolution profile are similar, and this is again the same requirement for US and Europe, it is known as profile comparison. In first example, we have seen that the 85% or more than the 85% release is observed in 15 minutes. But if it is not achieved, then you have to go for the profile comparison. And F2 calculation is required for test and reference product or between the test and reference product. And for calculation of F2, the 12 unit data is required. The mean of the 12 unit is considered in the calculation. So generate the dissolution of 12 units and use the mean data. Dissolution time point should be the same for the test and reference. And this is very obvious requirement that dissolution time point should be same. A minimum of 3 time points should be considered. This is a regulatory requirement and it is similar for US and Europe and many of the other authorities. You have to consider the 3 time points minimum. You can also consider 4 time points, but minimum should be 3. Then percent RSD or the coefficient of the variation should not be more than 20% at the earlier time point and should not be more than 10% at the later time point or other time points. And if your formulation shows less than 20% RSD in initial time points, for example 15 minutes, and less than 10% at later time points, then you can say that the product is not having more variability or it is a low variability product. The formulation is not showing variability into the dissolution. So F2 comparison involves a calculation based on log conversion for the difference of dissolution values between reference and the test. So this is reference and the test, and this calculation will give you the value from 0 to 100. If you get the F2 value of 50 or more than 50 for the test and reference profiles, then you can consider or you can say that the dissolution is similar. If the F2 value is less than 50, the profiles are considered as dissimilar or you cannot prove the similarity. The F2 similarity factor is a logarithmic transformation of the sum of squared differences. So this is sum of squared differences. This is the sum of these squared differences between the two dissolution profiles. That means reference minus test. F2 considers the average differences between the two profiles. It evaluates the similarity and differences in the dissolution profile. See here, you have to generate the 12 unit data and you will take the mean of that 12 units and then you will go for the F2 calculation. So F2 calculation considers the average differences between the two profiles. Now if the RSD is more, if percent RSD is more than 20% at the earlier time point like 15 minutes and more than 10% at the other time points, then it will go into a category of high variability type of formulation. And if the product is showing high variability, then the applicant cannot use the F2 similarity factor comparison or the dissolution similarity justification based on the F2 calculation. And this is why, because the RSD is more, and RSD is more because the unit to unit differences are high, and that's why the data is variable, and so the other techniques are used for this type of formulation. Then the F2 is not possible. Other methods when F2 is not applicable are bootstrapping method, that is bootstrap F2, and the MSD method. So this bootstrap F2 method and MSD method is used for the formulations which show high variability in the dissolution data. And high variability means more than 20% RSD at initial time point and more than 10% RSD at the later time points. So here I have taken an example. This is a hypothetical example. you can do the correct calculations for your formulation. Here I have put the data of dissolution for the reference product. 5 minutes 50%, 10 minutes 65%, 15 minutes 75%, 20 minutes 80%, 30 minutes 85%, then 90, 98. So this is a 60-minute profile, and here around in 30 minutes we are getting dissolution of 85%. Our earlier criteria was more than 85% in 15 minutes. So here you are not getting the 85% dissolution in 15 minutes, so you have to go for the profile comparison. Now test 1 data is here. It is giving you the F2 value of 80. It is similar. Then test 2, it is giving you the similarity with F2 of 56. Formulation 3 is giving you 67 F2. It is also similar. Then test 4, it is giving you the 100% F2. If the two profiles are exactly similar, then your F2 will be 100. Then if you consider the dissolution profile with slower release of around 10% slower release, it is not giving you the F2 of 50, and the faster profile also is not giving the F2 of 50. So it means that the F2 calculation considers the 10% difference, and if the difference is more than 10%, then you will not get the F2 value above 50 or equal to 50. So this is very simple. Your differences between the test and the reference dissolution profile should be within 10%. Then only you can get the F2 value between 50 to 100. So F2 similarity is the important criteria for showing the dissolution similarity. The percent RSD of the product should be less than 20% for the first time point and less than 10% from the second to last time point. So we will see the US requirement and the EU requirement for this F2 calculation now, because many of the people are confused between these two requirements for US and Europe. So as per US, the minimum of 3 time points are taken. Take time points from 15 minutes onward if needed. Then only one measurement should be considered after 85% dissolution of both the products. So if you are considering more than one time point after 85%, that means you are considering the similar time points or the similar near to similar results. That's why only one measurement should be considered after 85% dissolution. Earlier time points are the 5, 10, 15 minutes and should be less than 20%, and other time points percent RSD should be less than 10%. Now if we take time point as 5 minutes as a first time point, then it should have a less than 20% for RSD, and from second to last time point percent RSD should be less than 10%. Now coming to the EU requirement, a minimum of 3 time points are required, 0 excluded, and the first time point before 15 minutes, the second one at 15 minutes, and the third time point when the release is close to 85%. So compared to US, the Europe is giving you the correct requirement in words. They have written the words that 0 is excluded. In US, 0 is excluded also there. Then first time point before 15 minutes, you can take time point of 10 minutes. So 10, 15 and the second time point, one at 15 minutes and the third time point when the release is close to 85%. Take one time point before 15 minutes, not more than one mean value of 85% dissolved for any of the formulation, or that is the value of more than 85% dissolved for any of the formulation. But this is not the case with the US requirement. Here only one measurement should be considered after 85% dissolution of both the products. But here 85% dissolved for any of the formulation. If your test formulation is showing faster release and it is giving you the more than the 85% dissolution at 30 minutes, and if the reference product is not giving, but still you have to close with your test formulation which is showing 85% release. But this is not the case with the US. Then F2 similarity challenges and the limitations. So see, F2 calculation is always with the some challenges and the limitations, and F2 calculation is based on the average values of the 12 unit data. F2 similarity is achieved but bioequivalence failed. So can this happen? These are the some of the questions. So yes, F2 similarity may be achieved but bioequivalence may get failed. And why this happens? This happens because of the molecular behavior. We don't know how the molecule will behave in the in vivo condition. Sometimes faster profile may get passed into bioequivalence study, or sometimes the slower profile may get passed in the bioequivalence study. F2 similarity can be achieved by changing the dissolution parameters like media volume, RPM, and use of surfactants. So yes, if you are using the conditions in which you are not getting the F2 value, you can optimize the parameters or change the parameters, and then you can get the F2 value. But also remember here, just getting F2 value is not the requirement. With F2 similarity, the bioequivalence should be proved. So always bioequivalence study is done, and based on that bioequivalence study, the dissolution specifications are finalized, and then that batch dissolution will become the reference for the comparison for the other strengths and for the super. F2 failure can be justified with the clinical relevance or pharmacokinetics. So yes, F2 failure can be justified if you have IV-IVC data in your hand, and then you can say that, or you can justify that though the F2 is failing, but the clinical effect will not be significant or the pharmacokinetics will be same. Then how to deal with highly variable data? If your formulation is showing high variability, then you can go for the other methods like F2 bootstrapping and EMSD method. How to deal with the product with strength dependent dissolution? Dissolution cannot be compared as the lower strength shows faster dissolution. So as the higher strength will have higher dose and the lower strength will have low dose, so generally it is seen that the lower strength shows faster dissolution with the similar dissolution conditions as those used for the higher strength. So that time use of multiple units of the lower strength can be done, and the dissolution data is generated on the multiple units, and then dissolution similarity can be established and the comparison can be made. And why this lower strength shows faster dissolution? The reason is the saturation solubility based on the dose. So if you have a tablet with 400 mg dose with a higher strength, and you have 100 mg strength with the low dose, so directly there is a 4x increase in the dose, so the sink condition will vary, and that's why the lower strength will have faster dissolution. Another reason is the tablet weight or capsule fill content. Higher strength will have higher fill content and lower strength will have low fill content. And if you are going to check the dissolution of both the strengths with similar parameters, then certainly you will get the faster dissolution profile. and if you are getting the faster dissolution profile, then there are chances of the F2 failure. So in these cases, multiple units of the lower strengths are compared with the higher strengths. So this is regarding the F2 similarity, and I hope uh, you have got some knowledge out of this video for dissolution similarity. Always try to have dissolution similarity from the initial development, and always have the mild dissolution conditions and lower surfactant levels in the dissolution for F2 comparison. Also, multimedia multimedia dissolution should be done, and in multimedia dissolution F2 should be compared. Then F2 is also required to be compared for the alcohol dose dumping type of studies. So for extended release formulations, minimum 3-4 time points are required to be considered. If the extended release formulation is there, for example, and then you are going to compare the alcohol dose dumping data, that time also the dissolution similarity is required to be proved. So thanks for watching the videos from Pharma Learning in Depth channel, and I am very happy to see that many of the people are watching the channel video and they are getting benefit out of this. So stay tuned to Pharma Learning in Depth channel. Watch these videos and have good understanding and learning. Thank you.